So, Chapter 11, which is Introduction to Genetics, starts Unit 4, which is genetics. So, the next two chapters we do, Chapter 11 and Chapter 12, will deal with genetics. And we're going to start off with Mendelian Genetics, the work of Gregor Mendel. And in the next chapter, we'll move into molecular genetics, where we study DNA and RNA and proteins. Gregor Mendel was a monk who is considered the father of genetics. Genetics is the scientific study of heredity. Heredity is the information passed between parents and their children. So as information is passed from parent to children, that's called hereditary information. It's the product of meiosis, which we'll learn about in section 4 and 5 of this chapter. True breeding means that if allowed to self-pollinate, the resulting offspring would be exactly the same as the parent. All right, so what we have is when we call plants true breeding, that means that when they are self-pollinated, they end up identical. All right, and this is going to be an important concept when we deal with the idea of commercial plant growth. Cross-pollination, male sex cells in the pollen from one flower are used to fertilize the egg cells of a flower in another plant. And we can see that on this next slide right here, that we see the male parts of the plant with the pollen at the end. A little paintbrush is used to gather the pollen and place it onto the female pistil of another plant for reproduction to occur. Trait is a specific characteristic, such as color or plant height, that varies from one generation to another. When Mendel was doing his experiments, he called the parents the P and the offspring the F1, or first filial. Right. And we'll see that when we do our Punnett squares. It's important to make sure that you label your generations either P for parents or F1 for first filial, or F2, which we'll get to later. Hybrid is a cross between parents with different traits. All right, so you have these parents, they have different traits, and we're crossing them together. Mendel did 7 different crosses of 7 different traits. All right, and we'll see that on the next slide. In the crosses Mendel did, he found that the traits of only one parent showed, though. In each case, one trait seemed to be lost. So if we take a look right here at this, we can see the 7 crosses that Mendel did. And what we see is that in each case, we went from 14 characteristics down to only 7 characteristics. Okay, so we lost 7 traits, or Mendel lost 7 traits, and he didn't know where they went. Okay, and what we now know is it's the factors of dominance and recessiveness that caused this loss of traits. Right, they weren't lost, they were just masked by the dominant. The chemical factors that determine traits are known as genes. Genes are arranged in a linear fashion on the chromosome. Each gene has a specific location. The different forms of genes are known as alleles. Right? And we're going to deal with alleles when we start talking about Punnett squares. Okay, so there are the different forms of the traits. So you have tall and short, or in that last slide, the green or yellow. Those, you would have alleles for each of those. Okay. Each organism has 2 alleles that control every trait. Gene expression is how each trait is expressed within an organism. The principle of dominance states that some alleles are dominant and others are recessive. And Mendel wanted to know if the recessive traits had disappeared, so he had the F1 generation self-pollinate to form an F2 generation. Right? To explain why the recessive trait had reappeared in one situation, Mendel decided that the dominant allele only masked the recessive one. So that's what we were talking about. We have a masking of the recessive allele by the dominant one. Segregation is the separation of alleles. So alleles are segregated from one another during meiosis, getting ready to form the sperm or the egg. So the gametes are the sex cells. They're the sperm or the egg. So when they are formed, alleles are separated from one another. So each person has 2 alleles for a trait. All right? Say height. Big T for tall, little t for short. All right, so we'll use one letter for each. And then when forming the gametes, they're segregated, separated from each other, so each gamete only has one letter, so that when the sperm and the egg come together, the second allele comes from the other gamete. When each F1 plant flowers, the two alleles are segregated from each other so that each gamete carries only a single copy of each gene. Therefore, each F1 plant produces two types of gametes, those with the allele for tallness and those with the allele for shortness. So if we look at this picture on the next slide here, this picture shows that the F1, the parents in this case, well, the first generation of offspring, but they're going to reproduce, and we see that they have a big T and a little t, and when we form the gametes, they each have one letter. They're segregated, and now we can cross the two F1s, and we see the possible combinations. All right? And we'll do that more when we get into Punnett squares.